So, good evening to everyone and welcome to this video class. Myself, Dr. Narkede Gopal Vasudev. I'm working as Assistant Professor and Head, Department of Genetics and Plant Breeding, School of Agriculture, SR University, Warangal, Telangana State. So, today we will deal with crop improvement techniques in two most important solanaceous crops, that is chili and tomato. So first we will start with chili crop. As we are well aware, the botanical name of chili is Capsicum annum. Okay. So Capsicum annum L. So what is this L? L is the name on the name of scientist, that is Carlos Linnaeus. What he did? He gives taxonomy of plants. Divided the plants into different his name. Write L on after every botanical name, there will be L. So chromosome number of chili is 2n is equal to 24 and 2n is equal to 26. That is a haploid number is n is equal to 12 and n is equal to 30. The family of chili is Solanaceae. Okay. And chili is regarded as a self-pollinated crop, but the cross-pollination percentage in chili is almost 63%. That's why you can consider the chili as often cross-pollinated crop. Then the fruit in the chili fruit is called as paprika. The pungency in chili is due to capsaicin. Red color in chili, it is due to capsanthin pigment. And next is fruit type in chili, it is called as berry. And center of diversity of chili is Mexico. So this last 5-6 points are very important as far as your competitive examinations are concerned. Like paprika is the name for chili fruit. Then capsaicin is responsible for pungency in chili. Then next, capsanthin is responsible for red color in chili. Then fruit type of chili is called as berry. And Mexico is the center of diversity. So what is this center of diversity? Center of diversity means a center where you can find a maximum genetic variation. Where you can find a maximum genetic variation of that particular crop. So that center is regarded, then you can consider that center as a center of diversity for that particular crop. So in that case, Mexico is center of diversity for chili. So here you can see the diversity in chili and peppers. So here you can see different chilies with different shape, size, color, texture. So like different lengths are there. Some are circular in shape. Some are very thin. Some are red. Some are dark red. Some yellow, orange. So each and every size and each and every color. So the variation, lot of variation is there in chili crop. So here you can see easy. Okay. Yeah. So now we will move towards flower morphology. So in chili, the flowers are white with blue to purple color anthers. Calyx smooth to serrated. Stamens are alternate with the petals. Okay. So what is meant by petal? Petal is nothing but single member of corolla. Okay. Then stigma is mostly exerted, leading to high degree of cross pollination. I already told you the cross pollination percentage is almost 63 to up to 70%. So the flowers are actinomorphic, bisexual, pedicellate, and hypogynous. So what is meant by actinomorphic? So actinomorphic flower means you can divide that flower in two equal parts in any plane. That means vertical, horizontal, or diagonal cuts. If that cut will pass through central axis, so you can divide that flower into two equal parts. That flower is called as actinomorphic flower. Bisexual flower means your complete flower. That flower contains male and female parts. Pedicellate, flower with pedicel. That is a stalk-like structure is there. And hypogynous flower means the position of ovary is above the all remaining floral rows, right? So that means the ovary is superior. Ovary is superior to the remaining floral rows. Then next is gynoecium. So in Chile, gynoecium is bicarpellary syncarpus. Bicarpellary means presence of two carpels, and syncarpus means fuse. So two fuse carpel, that is called as bicarpellary syncarpus. Bi or tetralocular with superior ovary. Okay, so number of stamens are 5. Epipetalous condition of stamens. As I have already told you, stamens are alternate with the petals. Yeah. So in this slide also you can see variation in flower color, fruit shape, fruit size. That whole variation here you can see clearly. Yeah. So now you can see here some flowers are purple, some white, yellowish flowers are there. Anthers also yellowish to purple color anthers are there. So here you can see a proper flower and fruit variation available in capsicum. Yeah. So the next is isolation distance for the purpose of seed production. As we are already aware, there are 4 different classes of seed. That is nucleus seed, breeder seed, foundation seed, and certified seed. So in Chile, for breeder seed production, isolation distance is you have to maintain 500 meters. And for certified seed production, you have to maintain the isolation distance 250 meters for production of certified seed in Chile. So as far as bell peppers are concerned, for breeder seed production, isolation distance is 400 meters. And in case of certified seed production of bell peppers, isolation distance you have to maintain 200 meters. So what is this isolation distance? Isolation distance means if you are going for breeder seed production or certified seed production, you have to maintain a safe distance from the same crop field. There should be a 500-meter distance in between two chili plots for breeder seed production and 250-meter distance as far as certified seed production. So that distance is called as isolation distance. Yeah. So now we'll go through one by one the different capsicum species, available capsicum species, which are mainly used in crop improvement. So first is capsicum annum. So that capsicum annum we can consider as a cultivated one. So in that capsicum annum also fruit variation is there. So here you can see different nine different fruits are there available. Okay. So then second one is capsicum frutescens. Here also you can see the different fruits, right? So lots of diversity is available in capsicum. Yeah. So next is Capsicum chinensis. Here you can see different 11 fruit types are there in Capsicum chinensis. With shape, color, size, everything is different. Then next is Capsicum Galapagos. Small fruits, just like you can see the fruits like cherry tomato. Complete different structure. The next is Capsicum chacoense. And next, Capsicum scotianum. So in Capsicum scotianum, you can see the difference in flower. On corolla, some patches are there. And look at this fruit. Fruit is quite shiny. And small size fruit is there. Next, Capsicum baccatum. Again, in Capsicum baccatum, 7 different fruit types are there with different colors. Next, Capsicum pubescens, purple corolla. Next, Capsicum pratermissum. You can see one by one is different in flower structure and fruit type with fruit color also. Next is Capsicum eximium. So this is the diversity available in capsicum species. Okay. Then look at this. Next is Capsicum caldanaceae. So these are the some species available in capsicum. Yeah. So the most important is Bhuj jolokia. Bhuj jolokia, we can found this Bhuj jolokia in Arunachal, Assam, Nagaland, and Manipur. So actually, this Bhuj jolokia is a hybrid. So this Bhuj jolokia is a hybrid between Capsicum chinensis and Capsicum frutescens. Okay. So this chili hotness is measured in, what is the unit? SHU. So what is this SHU? That is Scoville heat unit. Okay. So what is the hotness in Bhuj jolokia? It is 1001304 SHU. That is 10,01,304 Scoville heat unit. Now we'll move towards some breeding aspects. That is, what are the breeding objectives of chili crop? So the first foremost breeding objective, higher yield, right? Farming any crop, if you grow any crop, so main ultimate aim will be higher yield, more pungency in hot pepper, zero pungency and more antioxidant as far as sweet pepper is concerned. Then next is, as we are quite aware, anthracnose is the most severe disease in chili, along with leaf curl and bacterial wilt. So you have to breed resistant cultivars. The next objective, more dry powder than green fruit. As we have already seen, the breeding objective, that is higher yield. So we need more number of fruits per plant. More fruit weight, that fruit size should be large. fruit shape, size and color should be uniform. Okay, and whatever the cultivars we will breed, either varieties or hybrids, that should be early mature. Wider adaptability, high nutritional quality, and the last but not the least, long shelf life, so that we can easily transport the product from one place to another. Right? So these are the some main breeding objectives in chili. So now the most important question arises: why there is a need to develop or to breed hybrids in capsicum? What is the need? So first is that we need some cultivars or hybrids or some progenies with earliness, and high yield. Then, as I have already told you, there should be uniformity in fruit size, shape, and color. Then, improvement in fruit quality. Okay. And next is wider adaptability. And if you will go with earliness, high yield, uniformity, fruit quality improvement, ultimately you will get high economic returns. So these are some 5 points which are responsible for hybrid development in capsicum. So now for seed production, you need to plant female to male ratio. Okay. So the ratio of female to male is 4:1 for seed production. Then what are the different stages of rogging? So what is meant by rogging? You can say removal of unwanted plants, removal of weeds, removal of off types, right? So what is meant by off type? So in off type you can say other crop plants, some volunteer plants, some chili plants, but phenotypically that should be different. Okay. So there are mainly 3 stages of rogging. First is before flowering, second after flowering, then at the time of fruiting, right? So the male sterility present in chili is genetic and cytoplasmic genetic male sterility. That means both GMS and CGMS are present. But though both male sterilities are there, now at commercial level, the breeders are mainly concentrated on CGMS system for development of chili hybrids. So fertilizer dose is 30-60-30 NPK kg per hectare. Seed is approximately 200 kg per hectare, that is 20 to 25 grams per plant. The next is what about seed rate? For varieties, 500 gram to 1 kg per hectare. And for hybrid, 200 gram for female parent and 50 gram for male parent. 45 by 45 centimeter is the spacing. Okay. So major diseases are, as I have already told you, anthracnose, leaf curl virus, and powdery mildew. Again, we will see the major diseases one by one in next slides. So this virus mainly transmitted by white flies. Then what are the breeding methods? In conventional method, introduction is there, pure line breeding is there, pedigree method is there, backcross method is there, heterosis breeding, and last one is instant hybridization. In modern methods, marker assisted breeding is there, tissue culture is there and mutation breeding is. So now commercially marker assisted backcross breeding is used for transfer of some desirable characters from different species, as I have already shown to you some Capsicum chinensis, Capsicum baccatum, Capsicum frutescens. So some desirable characters are transferred by using this marker assisted backcross breeding. Right. Yeah. So here we will see one by one different diseases. So first major disease is anthracnose, which is caused by a fungus Coledotrichum species. So here you can see in anthracnose, fruit damage is there. So here you can see the symptoms, right? As far as control measures, seed treatment with Thyrum or Captain, 3 to 4 gram per kg of seed. Then you can spray a liquid copper spray and sulfur powder weekly. Then 0.25% mancozeb and 1.5% Captain before and after flowering. Then last is neem oil spray every 7 to 14 days. So this is most severe disease in Chile, anthracnose, caused by fungus. Actually, it is a fungal disease caused by Coledotrichum species. So next is again one fungal disease, powdery mildew, caused by a fungus Libellula taurica. So powders, you can find powder on leaves, total plant. So as a control measure, use of garlic extract mixed with liquid soap. You have to spray once in a week. Then second, spray with wettable sulfur 0.3% or carbendazim 0.1%. Then third is damping off, which is caused by fungus Pythium myriotylum. Here, a total plant will damage, right from root to shoot. So to control this, seed treatment with Thyrum 2 to 3 gram per kg of seed. Apart from that, you can drench the soil by using copper oxychloride, metalaxyl or Bordeaux mixture. So by applying this, we can control damping off. So next is Alternaria leaf spot. Again fungal disease, that is Alternaria alternata. You can see the symptoms also. I have attached the pictures where you can clearly see the symptoms. So as far as the control measure, mulching. Mulching is most important because spores in soil cannot splash onto the leaves, right? And crop rotation with non-solanaceous crops. That is again most important. Crop rotation so that you can maintain the soil fertility also. So next is Cercospora leaf spot caused by Cercospora capsicum. Here also you can see on leaf spot, the symptoms you can see. So to control this, you have to do seed treatment with hot water at 52 degrees centigrade for half an hour. Then apart from that, second, you have to spray chlorothalonil 2.5 gram per liter or mancozeb or carbendazim. Okay, so these are the some important points as far as capsicum species is concerned. So now we will move towards our next crop, that is tomato. So botanical name of tomato is Solanum lycopersicum. Chromosome number is twice n is equal to 24, n is equal to 12. Family same Solanaceae. So origin South America and Mexican region. So in India, tomato is cultivated in UP, Karnataka, Maharashtra, Gujarat, Punjab, Haryana and Bihar. So immediate ancestor of tomato is Solanum lycopersicum variety cerciforme, sorry. Immediate ancestor is Solanum lycopersicum variety cerciforme, that is cherry tomato. So domesticated species is Solanum lycopersicum, ancestor is Solanum pimpinellifolium, and wild relative, closest to wild relative is Solanum peruvianum. So here, like I have shown in Chile, so here also you can see a diversity available in tomato crop. Some red, oranges, orange color, small fruits, different fruit structure, fruit type, color, you can see here. So now we'll move towards flower morphology. So 5 sepals with trichome. So what is this sepal petal? So single member of calyx is called as sepal, single member of corolla is called as petal. Right? Single member of androecium is stamen and single member of gynoecium is carpel. 5 sepals with trichome, 5 petals, bright yellow and gamopetalous condition, that is fused. Androecium with 5 stamens. Gynoecium superior ovate, again bicarpellary syncarpous. That is 2 fused carpel. So tomato is regarded as a self-pollinated crop. Almost 94 to 99% self-pollination is there. And 2 to 47% natural cross-pollination is reported based on temperature, plant height, plant population and the genotypic nature of that particular plants. So here you can see, so this is pedicel, this is corolla, this is calyx, that is sepal, right? These are the petals. Then this is stamen, this is stamen. Then style, stigma and ovary, this is ovule. Here you can see the dissection of fruit, that is seeds. So here you can clearly see the different parts of flower. All four roles are there: calyx, corolla, androecium and gynoecium. With transverse section of fruit, you can see here seeds also. Okay. So now we will see one by one tomato species. So botanical classification of cultivated tomato according to the fruit shape and plant spread was done by Bailey in 1949. So according to him, first is Solanum lycopersicum variety commune. You can see here, that is common round fruited tomato. Common round fruited tomato. Second one is Solanum lycopersicum variety grandiflorium. Solanum lycopersicum variety grandifolium, that is potato leaf style, right? Then third one, this one, Solanum lycopersicum variety cerciforme, that is small fruited. Then next is Solanum lycopersicum variety validum, that is upright tomato. And last is Solanum lycopersicum variety pyriforme. That is pear-shaped tomato. So these are the some types of cultivated tomato. So then some related species we will see. That is, first one is Solanum pimpinellifolium. Here you can see the picture of flower and fruit. So first species is Solanum pimpinellifolium. So this Solanum pimpinellifolium is a source of resistance for fusarium wilt and early blight. So here you can see the small size fruits. So the next is Solanum peruvianum, which is a closely related to cultivated tomato. So this Solanum peruvianum is resistant for leaf curl virus. Here you can see the fruits with some purple, green to purple color. The next is Solanum chismani, which is a salt resistant wild species. So here you can see the fruit color is different. Small fruit size is small, but fruit color is somewhat different. Then next is Solanum abrochates or Solanum hirsutum. Green fruit. So this Solanum abrochates or hirsutum is fruit borer resistant. Okay. The next is Solanum pennilli. So this Solanum pennilli is drought and salinity resistant wild germplasm. So in tomato also, like chili, a large amount of diversity is present. Then Solanum chilens, which is Gemini virus resistant wild germplasm. Yeah. So after seeing some diversity in tomato, now we will see breeding objectives. Right. So first is breeding for earliness. Second, breeding for increased fruit yield. Third, breeding for fruit quality traits. So now what are the fruit quality traits in tomato? Shape, then uniform size, deep red color with increased shelf life for transport purpose. Then next is breeding for disease resistance. So there are few severe diseases in tomato. So breeding for disease resistance is a primary breeding object, right? Then breeding for insect resistance, that is fruit borer and whitefly. That whitefly is responsible for viral disease. Then breeding for abiotic stresses, that is cold tolerance, drought resistance, salt, low temperature. As we have seen, some wild germplasms are resistant to drought and salt. So we can use that germplasm. Then prolonged storage and transportation and development of indeterminate cultivars for greenhouse production. So next is botany of tomato. That is first is determinate plant type. So what is meant by determinate plant type? In determinate plant type, the main axis terminates with flower cluster. Then second is indeterminate plant type. So mainly there are two types of plant types available in tomato. Determinate and indeterminate. So determinate means your main axis terminates with flower cluster. And indeterminate means inflorescence occurs at every third internode. Inflorescence, you can see inflorescence at every third internode and main axis continues to grow indefinitely. So optimum temperature for pollination is 21 degree. 60 by 45 centimeter spacing. So the flower is ebracteate, bright yellow, chasmogamous. So what is meant by chasmogamous? So pollination occurs in closed flower. So flower open after pollination. Actinomorphic, as I already told you, you can divide the flower in two equal parts through any plane. You can give the cut through central axis through any plane. You can divide the flower. Hermaphrodite, that is nothing but bisexual. Okay. So pollination success rate in tomato is 50 to 70%. Ideal temperature for seed set is 22 to 28 degrees centigrade. Inflorescence as basal flower sets it better. So the basal flowers will set seed better. Again, fruit is called as berry. So 6 to 8 weeks from first flower to ripe fruit. The duration is 6 to 8 weeks. 1000 seed weight of tomato is 3 to 4 grams. Then seed weight of 250 to 330 seeds is equivalent to 1 gram. Right? So these are some basic informations. Then season for tomato is May, June for rainy season, and for post rainy, or you can say intermediate in post rainy and summer, so you can go for November to December. Right? So inflorescence is called as cyme. So flower is, as I have already told you, perfect and hypogynous, that is superior ovary. Tomato is warm season plant. So warm sunny weather is very well suited for ripening, color development, quality and high yield as far as tomato is concerned. Then soil type. Sandy loam soil with pH 6 to 7 is suitable for tomato. Seed rate 500 to 800 gram per hectare. So how many seeds will be there in 1 gram? And we have already saw, that is 250 to 330 on an average, 300 seeds. So again, rogging means removal of unwanted off-type plants. Okay. So three different stages, that is pre-flowering stage, flowering stage and fruiting stage. Ratio of male to female is 1:5. Right? Emasculation done on 55 to 65 days after sowing. So isolation distance for foundation seed production is 50 meter, and for certified seed production is 25 meter. Germination percentage in tomato is 70%. Seed is in open field 50 ton per hectare, and in greenhouse or glass house 150 ton. So now what are the breeding methods in tomato? That is same: introduction, pure line, single seed descent, single plant selection, mass selection, pedigree, backcross, heterosis breeding, interspecific hybridization, mutation breeding, polyploid breeding. Okay, these are the some breeding methods. So the detailed about breeding methods we will see in our next videos. We will see one by one. We will take one by one breeding method in detail with examples. That is case study. So now we will see major diseases of tomato. That is first one. Damping of which caused by different fungi. Pythium, Phytophthora, Rhizoctonia and Fusarium. So what are the controls? Soil drenching with copper oxychloride, 50% vegetable powder, 2.5 gram per liter. Right. Then seed treatment with metal exit, 24 hours before sowing. Then apart from both, seed treatment with Trichoderma aspergillum at 4 gram per kg or Bacillus subtilis at 10 gram per kg of seed 24 hours before sowing. Soil application of Bacillus subtilis at 2.5 kg per hectare. So apart from all this, again soil application of Bacillus subtilis at 2.5 kg per hectare with 50 kg of farmyard manure. So all these are controlled for damping off, right? So next is septorial itch spot caused by fungus Septoria lycopersicum. So as far as control measures are concerned, first you have to remove and destroy all the affected plants or plant parts, right? Then seed treatment Trichoderma asperellum at 4 gram per kg. Then fluxapyroxide at 250 gram per liter plus pyraclostrobin 250 gram per liter. Okay. So these are the control measure for septorial leaf spot. Then third is bacterial stem and fruit canker. It is actually a bacterium, Clavibacter michiganensis, subspecies michiganensis. So here you can see the symptoms of different diseases I am telling you. So here simultaneously you can see the symptoms in the attached pictures. So for control measure of bacterial stem and fruit canker, seed treatment in hot water at 50 degrees centigrade for 25 minutes. Then again seed treatment with mercury chloride 1:1000 is also recommended for the control of this disease. Then crop rotation with non-host crop, that is non-solanaceous crops, will lead to reduction of this disease incidence. Then you can soak this seed in streptocycline solution with a dose of 1 gram per 40 liter of water for 30 minutes. So that will protect the seedling in the initial stages of growth. In initial growth stages only, if you soak the seeds in streptocycline solution for 30 minutes. So next is early blight caused by fungus Alternaria solani. So as far as the control measures, first you have to remove and destroy the affected plant parts. Then second, crop rotation. Third, spraying of the crop with difolaten 0.2%, then diten M45, then bavestin is recommended for effective disease control. Then next is bacterial leaf spot caused by bacteria, that is Xanthomonas species. So this is the difficult to control once it appears in the field. Okay, so you have to use the disease-free seedling, and crop should be rotated with non-host crop. Then seed treatment with mercury chloride 1:1000. Seed treatment with mercury chloride 1:1000 is also recommended for the control of this disease. Then spraying with combination of copper and organic fungicide in a regular preventive spray program at 5 to 10 days interval, or spraying with agrimycin 100 ppm thrice at 10 days interval. Effectively you can control this disease. Right. Then the next is bacterial wilt. It is also one of the severe disease which is caused by bacterium Ralstonia solanacearum. Here you can see simultaneously you can see the symptoms on plant as well as on fruit. So the foremost important control is crop rotation. Then second, seed treatment with streptocycline 1 gram per 40 liter of water for 30 minutes. to protect at initial stages, right? So crop rotation and treatment with streptocycline so that we can control this bacterial wilt. Then next is leaf curl virus. Here you can see the symptoms on fruit as well as on leaves. So as far as control measure, soil application of granular insecticide like Puraden at the time of sowing in nursery beds. Then another dose of Puraden is given 1 week after transplanting and 2 to 3 foliar sprays with dimethoate or monocrotophos at 10 days interval controls the whitefly population because this leaf curl virus is a type of Gemini virus which can spread through whiteflies. This TLCV incident can be reduced drastically by the use of polythene mulching in the soil just before the transplanting of tomato. So this combined application of polythene mulching and Puraden application in the soil is recommended to control this leaf curl virus. Then next is tomato mosaic virus. So it is a type of Tobamo viruses. So as far as the control measures, first to foremost you have to select the disease-free seedlings or disease-free seeds, means the seeds which you are harvested from disease-free plants. So you have to select for sowing. So with seed treatment, soaking of seed in a solution of trisodium phosphate, that is 90 gram per liter of water. You have to soak the seed in trisodium phosphate a day before the sowing. Okay, the seed should be thoroughly rinsed and dried in shade. In nursery, you have to remove the infected plants and destroy it. That seedlings with viral disease should not be used for transplanting. And then next control measure is crop rotation. So these are some control measures for TMV virus. Then next is tomato spotted wilt virus. It is also viral disease caused by thrips. As far as control measure, the affected plants should be removed and destroyed. Alternate and collateral host. Harboring the virus, if some collateral host is there nearer to your plot, so immediately you have to remove at the time of weeding or earthing up operation to minimize the spread of this disease. Then 2 to 3 foliar spray with dimethoate 0.05% or monocrotophos 0.05% at 10 days interval. Control the thrips population. The next is fusarium wilt. You can see here the effect of this fungus on plant as well as on fruit caused by Fusarium oxysporum. The control measure is for the inspection of nursery for wilt infected plants. Then the affected plants you have to remove and destroy immediately. Then prior to planting, the bed should be drenched with carbendazim 0.1% and seed should be treated with thyrum, that is 2.5 kg per hectare. Then crop rotation with non-host crop such as cereal helps to reduce the disease inoculum. Okay, so these are some diseases we have seen. So now seed extraction methods. So there are three seed extraction method in tomato. That is fermentation method, acid method, and alkali method. Right. So here we will see one by one. So in fermentation method, you have to mix the fruit pulp with water for 24 to 48 hours. Okay. What are the silent feature? It includes low cost, no skill is required, but more time, low seed recovery, that is 0.5 to 0.6% only. The seed color will be dull and may be prone to seed-borne pathogens. Okay. So then second type is acid method. So in acid method, hydrochloric acid or sulfuric acid will be used at 10 ml per kg of pulp. You have to mix this fruit pulp with hydrochloric acid at 10 ml per kg for 20 to 30 minutes. As it requires acid, the cost is more and skilled labor are required for handling of acid. Time will be less. Seed recovery will be high, 0.8 to 1%. The seed color will be bright. So bright seed color means market value will be high. So with that acids, your soil-borne pathogens will be removed. Okay. So the care should be taken that improper washing leads to seed injury. Then third method is alkali method. Washing soda at 900 mg per 4 liter of water. You have to soak this fruit pulp in washing soda overnight. Okay. So in alkali method, seed recovery percentage is 0.7 to 0.8%. The seed luster will be lost in alkali method. And same, you have to take the care. Improper washing leads to seed injury. So here you can see some pictures of fermentation method, that is manual crushing, then fermentation, then washing of seeds. Here at last you can see the extracted seed material, right? Then in acid seed extraction method, this is mechanical crushing first, then extracted seed, acid treatment, and then washing of seeds. So this is stepwise extraction of seed in tomato. So this is my personal profile, as I have mentioned in my earlier videos. You can contact me on my given mail ID. And please like, share, subscribe and follow my LinkedIn page, YouTube page. And I am running one newsletter. LinkedIn page is there, that is Durja Agricultural Digest. Here you can get updates on crop productions, flower seed production, cereals, some genetics part, some genetics, breeding, crop improvement, crop production related. Here you can get the updates. So like, share, subscribe, and don't forget to follow. Okay. Thank you. Yeah. So thank you for watching my video on crop improvement techniques in chili and tomato. Hope you like that video and the available information shared with chili diversity in tomatoes, some diseases, major diseases in chili and tomato, with flower morphology and other information. So the next video will be on breeding for trait improvement in okra. As we all know, okra is a very important vegetable crop. So my next video class will be on breeding for trait improvement in okra. So as you can see, a great diversity is present in this Abelmoschus. This Abelmoschus genus is having a very high diversity that is available in India. Okay, so we will deal with this in detail, like flower morphology, breeding methods, some breeding objectives, with some case studies, how this cultivated okra and how this we can go with wild hybridization program, is which species are which species are crossable with this cultivated okra. Then again, genetic male sterility is there in okra. So how we can go for this breeding for development of hybrid based on this genetic male sterility. So all these points we will discuss in this video. So stay tuned. This full video will coming on 11th of October on my YouTube channel. So stay tuned and thank you. Thank you very much. Thank you very much for listening this. Thank you.